Dr. Cyrus Zabetian. Did I do it right, I hope? And he's here with us from, he's a professor of neurology, and he's here from the University of Washington and the VA of Puget Sound. He's also the co-founder and current director of the Washington State Parkinson's Disease Registry.
Great. Can you hear me in the back? Yeah, thumbs up, thumbs down? Better? I think that thumb's going up.
Close. Hold it really close.
Okay. I'm a bit myopic, but I think those thumbs were up. So I'm going to talk today. I'm a neurologist and neurogeneticist. And our task today was to talk about things that were new and exciting in the research world in this past year or so. Yep. I'm about to swallow it. Okay. Can everybody hear me now? All right. So again, our task today was to talk about some of the most exciting things that have happened in the world of research in Parkinson's disease over this past year or so. And there were many, many things that happened, but I'm going to focus on one specific topic, and that's precision medicine. So what is precision medicine? Precision medicine is the idea that if you look at a patient population, this could be any disease. And the idea is that everybody who has a single disease isn't the same. There are subtypes of diseases. And why does that matter? It matters because, for instance, if you can't distinguish these different subtypes and you give an entire group a treatment, that treatment might be very effective for a small subgroup of people. It might be extremely effective in 20%, but the rest of the 80% of those folks may have very little benefit or side effects, and you've really wasted resources and time by treating people who were unlikely to respond. Well, it sounds great. Why don't we just sort all this out? Well, the idea is that we have to have markers or a way of picking these different subtypes out. So when we do that, and if we're able to do that, what we can then do is that we can group all those people together. So all the people in blue here would respond to treatment A, and they'd respond to treatment A very well, and not to any of the other treatments. And if we can stratify all of these patients into the right groups, and then target a treatment that works just for their group, we've saved time, we've saved effort, we've made patients much happier, and this sort of theme can potentially hold true for any sort of disease. But there are areas where this has been in practice for years and other areas where it's just coming to bear. And the best example of this is cancer. So in many different types of cancer, and I'm just using breast cancer as an example, you take a piece of tissue and you look for receptors that are expressed on that cancer tissue. And so, for example, if your cancer is estrogen receptor positive, you get a different treatment than if it's negative. If you have human epidermal growth factor receptor, HER2, you get a different treatment. And these treatments are targeted for these particular receptors. So these receptors live on the cancer and make it grow. And so you will get, depending on which receptor you have, you'll get targeted treatments that bind to that receptor and shut it down. Okay? And so this idea has been going on for years in cancer treatment. But for diseases like Parkinson's disease and Alzheimer's disease and ALS, it's been a hope that we'd eventually be able to bring this sort of more precise approach to treatment, but it hasn't really happened until now. And so what's happened in the past couple of years is that we've started to be able to understand how we can start sorting out these groups. And how can you sort out groups? Well, there's lots of ways. So genetics is one of them. We can look to see if certain people have mutations in certain genes. There's lots of other ways. So in theory, we may be able to differentiate different types of Parkinson's disease by proteins you have in your blood or your spinal fluid. We might be able to do this by looking at things that you've been exposed to, your imaging, so MRI or PET studies. There's a lot of different ways that we could, in theory, do this. I'm going to simply focus on one area because that's where I think we have the most traction right now, and that's genetics. So many of you have heard of the GBA gene. Actually, I'd like to show of hands. How many people have heard about the GBA gene in Parkinson's disease? Just give me a raise. Just raise your hand if you have. So a few people, not too many people. So the GBA gene encodes this enzyme, glucocerebrosidase. And if you get a copy from your mother, copy from your father. If you get 2 defective copies, you get this disease, Gao Shay disease, which is a disease where an abnormal lipid builds up in cells all over your body. It makes your spleen large, it makes your liver large, it can crowd out your bone marrow, cause bone pain, cause your blood cell counts to go low. If it's a severe form, it can even cause death in utero or as a child. And a small subset of those folks get Parkinson's disease. But what we and others have found over the years is that if you have 1 defective copy, and people in this group who have 1 defective copy are much, much, much more numerous than these folks, it increases your risk for Parkinson's disease. And there are over 200 mutations in the gene. Some of them knock down the function of the enzyme a little bit, some of it a lot. So if you have a mutation that knocks it down a lot, you are even more likely to get Parkinson's disease. If you have a defective copy that knocks it down a little, you're at risk, but less so. Important to remember that just because you have one of these mutations doesn't mean you're going to get Parkinson's disease. It just increases your risk. There's probably other things that are going on with your lifestyle and your environment that combined with one of those mutations pushes you over the edge and gives you Parkinson's disease. So this enzyme, to get down to the weeds now, this enzyme, glucocerebrosidase, does one thing, and that's that it takes this compound, glucoceramide, which is a lipid basically that's in the plasma membrane of our cells all over our body. It's something we need, we have, it's part of our bodies. And it breaks it down into, it takes this glucose group off and creates this other molecule, ceramide. So when you have a defect, when this enzyme is defective, what ends up happening is this. It's just like when your sink starts backing up. This glucosylceramide molecule starts building up, and it starts building up, and it becomes a problem for your body. And the idea is that, and the idea is, here's that molecule. As this molecule starts to build up, there's something called a lysosome, which is an organelle in all of your cells that's essentially there for waste removal. It gets rid of proteins that have gone bad, and this is constantly going on all the time in all of our bodies. When this molecule, glucosylceramide, starts building up, it starts gumming up the lysosomes. They aren't working as well, and so other proteins start building up. That's the theory. One of those proteins that starts building up is alpha-synuclein, and you'll hear more about that in a little bit. But the idea is that it's this glucosylceramide building up that's causing a lot of the problems, eventually causes nerve cells in your brain to be sick like dopamine neurons and eventually die. So what can we do about this? So the difference here is that because we've known about Gao Shay disease and that it's a genetic disease for decades, there's been many decades of research on it. So we have a head start. And so there have been a number of different things that you can do you can replace the enzyme for people with Gao Shay disease, because you're mostly interested in what's happening in other areas of the body. But with Parkinson's disease, you can't just give somebody new enzyme because it doesn't cross into the brain. So you need to come up with other molecules. So here's an example of 3 clinical trials that are going on or have recently finished. So one of them is from Sanofi Genzyme. It's a glucosylceramide synthase inhibitor, and so it maps onto the map here. So if this is glucosylceramide, one of the things you could do to get it down is to actually hit the enzyme, inhibit the enzyme that makes it in the first place. So if you hit this enzyme, you can decrease the amount of glucosylceramide you have, and in theory, try to get things back in balance. So this has been in a phase 2 clinical trial that's ongoing right now called MOVES PD. I believe the Booth Gardner Center is a site for that study, as well as OHSU. The second compound is called LTI-291. This is an effort between Allergan and LTI Therapeutics. And these drugs essentially activate glucocerebrosidase. So one of them actually binds the good copy of the enzyme and increases its activity. The other acts as a chaperone, and that's this drug here, Ambroxol. I'll talk about that in a minute. But the idea here is that it's revving up the good copy of the enzyme and causing it to over-synthesize and overact. And then this drug, Ambroxol, is a drug that's been around for decades, and it was a drug that's been used for respiratory disease to thin your mucus. And people found surreptitiously that it seems to have an effect on glucocerebrosidase by taking the mutated enzyme and chaperoning it or following it around and making sure that it doesn't get broken down as fast as it otherwise would. And this is a drug that's off patent and is being studied by a number of investigators. So here you have targeted therapies that are really meant to see if the first studies are going to be to see if they work in people who have these mutations. And LARC2, probably more of you have heard of LARC2. Let's have a show of hands for LARC2. Okay, so quite a few more of you have heard about LARC2. So LARC2 is a gene that we found not because it was associated with another disease, but because of its association with Parkinson's disease. So there are several mutations in the gene. This is the gene. There are several mutations that have been described. A couple of them actually were first described in families right here in Washington State. And the most common mutation is this G2019S. And essentially, this curve simply is here to show that if you carry one of these if you carry the G2019S mutation, you have somewhere between a 25 and 40% chance of getting the disease by the time you reach 80, which means that the majority of people who have this mutation and live to that age don't get it. And again, why? It's probably because of a combination of other genes and things in your environment that we don't entirely understand yet. So the idea here is that this gene, there are different mutations, but what they all seem to have in common is that they affect this part of the protein, which is a protein kinase. And protein kinases basically go around adding phosphate groups to proteins. And why is that important? Because that's the way that proteins often signal each other to turn on or turn off. So by adding that phosphate group to a bunch of other proteins downstream, you can start causing all kinds of other proteins to do things. And if you overactivate or underactivate some of those proteins, you can cause a number of things to happen. And the idea is that nerve cells are particularly vulnerable to these actions, and so they take a hit. And if they go below a certain level, of course, you get Parkinson's disease. So here, most of the research has been trying to find small molecules that will turn down that kinase activity. So it's still working, but it's not adding phosphate groups as overactively as it once was. And there's one study that was just completed, a phase 1 study. This was just in healthy subjects. The company is Denali Pharmaceuticals, and this DNL201 is a small molecule that does just what I said. And this is a study essentially where the phase 1 studies, you hear us talk about phase 1A and 1B and 2 and 3 studies. So these early phase 1 studies essentially are there not to see if the drug actually slows down Parkinson's disease, but what the properties of the drug are like oftentimes in healthy controls. So these are just some pharmacokinetic data showing how long does it take if we keep giving you the drug for it to reach steady levels in your blood. Does it get into spinal fluid? If so, how much? Does it seem to bind to the molecule, so-called target engagement? Does it bind to the right molecule, and does it seem to cause any changes in that molecule? So that study was completed, and now a phase 1b study is just underway. So we know that this doesn't look like it harms people, and the next question is it going to actually do anything beneficial for the disease? That's an oversimplification, but. And I just want to point out that what's exciting about these drugs is that they are going to the heart of the problem. They are going into the cell at the molecular level, and in theory, hopefully, addressing the root of the problem. And so, and hopefully, if that's the case, they're not just going to treat the symptoms of Parkinson's disease like levodopa or dopamine agonists. They're actually, we hope, going to slow down the disease. This is just a diagram that sort of shows what we think happens, again, an oversimplification, when these are dopamine neurons, and as with normal aging, we all lose dopamine neurons. And if we start losing them too quickly, at some point, we get to the point where we start having early symptoms and then obvious symptoms, and then our disease gets very severe. The idea with neuroprotection is that you slow this process down. So if you catch it early, you may be able to delay the time at which somebody actually gets symptoms. And if they already have symptoms, you might slow how fast their symptoms get worse. And that is the holy grail of all neurodegenerative disorders. We don't want to just treat the symptoms. We want to prevent them or slow it down. And that's what people mean by a quote-unquote cure. So one last point that people often ask. So how many people does this affect? And I have this pie diagram here to simply show you that. So if you were to take a random sample of people across the US who have Parkinson's disease, about 1% of them are going to have a Lark2 mutation. About 8% of them are going to have a mutation or other variant in GBA that matters. So that's about 9% of people that these studies are going to pertain to in theory. So if they work, they could help about 9% of people with Parkinson's disease. Now, it's possible that these molecules will actually help people who don't have one of these mutations. We don't know yet. The studies are being initially targeted to people who do, but it's possible that they might help people who are part of the rest of the pie. And that would be wonderful if it happens. We just don't know yet. And I'll end by going right back to the beginning, which is, here we go with this. As I mentioned before, we want to stratify people based on a subtype of the disease, and we can do that a number of ways. And perhaps we've reached the point in Parkinson's disease where Group A is defined by a GBA mutation. Group B is defined by Lark2. There's another mutation in another gene that causes early onset Parkinson's disease. Perhaps those people will be a different group that respond to a specific targeted therapy, and on and on. And it isn't, again, that these different groups have to just be defined by genetics. We know that probably in a lot of people, genetics plays a role, but not an enormous role. So there are other markers that we need to and are continuing to look for to be able to stratify these groups and target the treatments to the groups where they would really do the most good. Okay, so with that.